recording. And essentially what we're doing is wrapping up chapter 8. And what we did last time with chapter 8, we talked about mutations, and we talked about bacterial diversity. And we said there are two ways in which bacteria can create strains. But to create strains, they have to change little parts of the genetic material. They have to acquire genetic new genes. So there's two ways in which they can acquire new genes. They can mutate and pass the mutations from mother to daughter, or through horizontal gene transfer, which means to exchange genetic information between two quote-unquote adult cells. So we talked about mutations, and we said there are two types of mutations. There are spontaneous mutations, and we have induced mutations. Spontaneous mutations are mistakes by DNA polymerase. And because DNA polymerase does not make mistakes very often, these are extremely rare events. The other mutations come from mutagens. And mutagens can be chemicals or radiation. And these are not rare events. They are a lot more common by factors of 1,000 to 100,000 compared to spontaneous mutations. So they may be 1,000 to 100,000 more common than spontaneous mutations. We talked about the different types of spontaneous mutations. We talked about silent mutations, missense mutations, and nonsense mutations. And we talked about frame shift insertions and frame shift deletions. We talked about transposons a little bit, and now we're into induced mutations. So induced mutations can either be due to a chemical or to radiation. And this is the range of rates of mutations, 1 to 1,000, 1 to a million. So they are a lot more common. So it's a lot more likely to create a mutation if you use a mutagen than if you just wait for the DNA polymerase to make a mistake. That's essentially what that says. Give me a second. I thought when it comes to chemical mutagens, it says here that there are two ways in which they act. There's really three ways. I need to one of these days fix this PowerPoint. A chemical mutagen could physically alter the nucleotide so that now the nucleotide has been changed and it will not bond correctly. So that now we can have an altered A that bonds with C's, for example. Another way in which a chemical mutagen can cause mutations is because they resemble nucleotides. So the mutagen itself will be incorporated into the DNA because DNA polymerase thinks is a nucleotide, but it's not. It doesn't base pair the way a normal nucleotide base pairs. The third way, which I don't have it here, is what we call intercalated agents. When the mutant, the chemical intercalates, inserts itself into the DNA. And this is different from the second way in that they're going to intercalate themselves not as nucleotides, but as molecules that are going to completely distort the DNA. Okay, and we'll go into explanations on that. Radiation is straightforward. We have two types, gamma rays and UV lights. Gamma rays oxidize DNA, and they're going to break DNA and cause damage. Thymine dimers is what UV light creates. Okay, so I guess we're going for chemical mutagens first. Okay, so that's a summary of how mutagens can act. All right, chemical mutagens First way that they act is that they alter DNA. So, for example, nitrous acids are going to modify Cs to act like U's, like uracil. So even though this is DNA and DNA shouldn't have U's, the C is now going to act as if it was a U. U base pairs with an A, C base pairs with a G. So now you're going to have a G of C base pairing with an A. Okay, so that would cause a mutation. Sodium nitrate is used to preserve meats like hams and bacon, and in the liver it is converted to nitrous acid, which is the mutagen. So that's why nowadays they have uncured meats that do not have these sodium nitrates. Alkylating agents are going as another type of these nucleoside altering mutagens, which are going to add alkyl groups to purines and pyrimidines, and they're going to begin to alter their bondings. Nitrosoguanidines is another one of those that add methyl groups and are going to again make guanine base pair with a T. So a G is now base pairs with a T. So it acts as if it was a C, because G's are supposed to base pair with C's. Okay, so that's essentially how these chemicals work. No, methylation should not cause mutations. No, methylation is something that the organism does purposefully. No, methylation affects both prokaryotes and eukaryotes. Yeah, it's in both. Actually, humans, we methylate also. But in our case, methylation could cause mutations. In the case of bacteria, methylations are done on purpose to brand the DNA so that the bacteria knows this is their DNA. But in humans, methylations could cause mutations, absolutely. So that's, yeah. And that's, again, something that we're learning with epigenetics. There's some chemicals, some things that we can be exposed to that would methylate DNA and could cause mutations in our DNA. So those are the base-altering mutagens. The base analog mutagens, or the ones that resemble nucleotides, are incredibly useful. These are used as antiviral and antiprotozoan drugs. So these are chemicals that look like nucleotides. DNA polymerase confuses them with nucleotides and incorporates them into DNA or even RNA if it is an RNA polymerase. But instead of continuing with the replication, they halt replication, or they cause point mutations because they're not going to base pair like a normal nucleotide will. So examples, a lot of the antiviral drugs are going to be nucleoside analogs. The first 8 drugs that we work with, like ACT, is a nucleoside analog. 5-bromouracil, which is a chemical used in research to stain DNA, and it's a pretty bad mutagen, takes the place of a T, but pairs up with a G. 2-aminopurine takes the place of an A, but base pairs with a C. Again, they look like nucleotides, they don't behave like normal nucleotides. And the importance of these kinds of mutagens is that we do use them as drugs. Okay, so this is a normal nucleobase, normal thymine. There's the base analog 5-bromouracil. And that's just more of the same. Okay, the third way in which these mutagens could work, drugs, mutagens, not that I know of, like 5-fluorouracil. You know what? They probably are. That one, the one you're mentioning, rings the bell as a mutagen. So they probably are. Another way they're used for is also in cancer drugs. So yeah, it's a pyrimidine analog. And, yeah, it's a cancer drug. There we go. Yeah. So a lot of the chemotherapy drugs are nucleoside analogs. Yep. So yes, that's how they work. Absolutely. Yeah. And we do know they're mutagens. That's what we're using them. We're trying to kill the cancer cells. So that was the second way that these things can work, these mutagens can work. The third way that they can work is as intercalating agents. And this is different in that these agents do not look like nucleotides. These are simply large molecules that are going to insert themselves inside the DNA. They're going to distort the DNA. And now when the DNA is replicated, this entire set of sequences are going to be left out. They're going to be skipped, causing tremendous mutations. This will be very bad deletions because we're not deleting one nucleotide, we're deleting many nucleotides. So there's no way that genes can survive those kinds of mutations. So they result in nonsense mutations that, if nothing, obviously knockout mutations. So examples, aflatoxin, ethidium bromide, another research compound, chloroquine, which is the anti-malarial drug. The good thing about drugs like chloroquine, though, is that they have been modified to recognize the protozoan DNA. So it's not as bad to the human DNA as it is to the protozoan DNA. It's still toxic. I mean, it's not a completely innocuous drug, but it's not as bad as it sounds. It has more selective toxicity. It has been made to have selective toxicity. Okay, so would mess with it so that it's safer now than it was originally. Aflatoxin is a different story altogether because this is something that we don't have much control over. Aflatoxin is a toxin made by a fungus called Aspergillus flavus. And the problem with this fungus is that it is everywhere. And it lives in symbiotic relationships with some foods like corn, peanuts, wheat. So it is impossible to get rid of Aspergillus flavus. It is impossible to get rid of aflatoxin. So all corn, all wheat, all peanuts are infected with aflatoxin, which is a intercalating agent, is a mutagen. So what we do is we have minimum levels that are acceptable of this toxin. And so I got this from the FDA website. So for example, all products except for milk, and all products will mean typically anything that has corn or wheat or peanuts, it is acceptable to have 20 nanograms, which is minuscule per gram. So a nanogram is 100,000ths of a milligram. So tiny, tiny amounts. The only thing, though, is that when it comes to animals, we allowed a lot more of aflatoxin. So the corn that is given to animals, for example, cottonseed, that's another one that has a lot of aflatoxin. So we allowed a lot more aflatoxin in these foods that are given to animals than what we consume ourselves, but obviously we're going to consume the animal eventually. So again, nothing we can do about it. Pretty much all grains have some form of aflatoxin in there. So it's just a matter of, just try to, make up by living healthy lives in other aspects, because there's no way that stop eating wheat, corn, peanuts, those three for sure. The first three, and animals, I guess. I don't know what we're left with, but okay. So once in a while, products like peanut butter may get recalled because they have too much aflatoxin, for example. Okay, so radiation, again, ionizing versus non-ionizing, gamma versus UV light. UV light is going to cause the thymine dimers, which distort the DNA and cause mutations. Okay, so that's pretty much it for that part of the chapter. Yeah, this is another nucleoside analog, aciclovir, which is or Sovirax, which is the drug that we give for herpes. Yeah, it's another nucleoside analog. Oh, Ames test. Okay. All right. So this is the deal. We talked a lot about mutagens, and mutagens are potentially carcinogens. So a mutagen could potentially lead to cancers. So wouldn't it be nice to be able to identify potential mutagens quickly, easily, and cheaply? So in the 70s, a doctor named Ames came up with a way of using bacteria to try to identify mutagens. And he created a very cheap, quick, and effective test, because normally you would have to use cells, cultures of eukaryotic cells, human cells, from there move on to animals, and which was a very expensive, time-consuming research. With these type of tests using bacteria, we can screen for chemicals that could potentially be carcinogenic, and if they come out to be positive, then we can take those positives towards the more expensive tests. So the Ames test uses a bacteria called Salmonella that is heavily mutated. And one of the mutations that it has is that the bacteria has lost its ability to make an amino acid called histidine. So we call those organisms histidine autotrophs. They cannot make histidine, which means unless we give it the histidine, the bacteria dies. So these heavily mutated histidine autotroph bacteria has to be grown in a histidine-rich media. If you don't give it histidine in the medium, it's going to die. Okay, so no, I'm not really going by what the PowerPoint says. This is the way it works. Let's see. What we do is we grow the bacteria. We have the bacteria in the histidine positive medium. Over here, this is the control. I'll go back to the control in just a second. We expose the bacteria to the chemical, let's say chemical X. Okay, I don't like this picture. Let me go ahead and start here better. It's better yet. Okay, so we have the bacteria in a test tube. And we expose the bacteria to chemical X. So we can have chemical X to the bacteria. After exposing to chemical X, we're going to grow the bacteria, there we go, that right there, into two plates. One plate has, let me just make sure I get which plate is this. Most remain histonegative and no growth. It's not, oh, never mind. I really don't like this picture. Okay. They're just confusing. Give me a second. Let me get myself my own little whiteboard. Okay. So we have the little bacteria in a test tube, mutated bacteria. We expose it to the chemical X, and the question is it a mutagen? All right, so we are going to, and mind you, this medium is histidine positive because the bacteria cannot make histidine, so we have to feed it to it, we have to give it to it. So now we grow the bacteria after being exposed to the chemical, we grow it in a medium that has no histidine. So the idea is, if the microorganism experiences mutations due to chemical, some of those mutations may revert the histidine negative mutation. So the microorganism is histidine negative, is a mutant. We expose it to a chemical that is going to cause so many mutations, maybe, that some of those mutations will revert the histidine negative mutation. So we put it in a medium that has no histidine, and only those cells that mutated due to the chemical and are now histidine positive, so these are going to be revertants, are going to grow in the medium. So let's say that 20 cells, 20 colonies, I should say, not cells, grew in the medium, which represents one cell. Each one of them came from one cell. So 20 colonies grew in the medium with no histidine. So that means 20 revertants. So the question is, okay, so is that a significant number? What would have happened had we just not exposed to the chemical? How many revertants would normally have appeared by spontaneous mutations? Okay, so we're going to do the same thing, but no chemical. We're going to grow those cells in a histidine negative medium and see how many grow that spontaneously revert by a spontaneous mutation. And let's say 2 colonies appear. So this is the control. So what this is saying is, by just spontaneous mutations, normal mistakes in DNA replication, maybe 2 cells out of millions, hundreds of millions, may revert and go back to being histidine positive without exposure to a chemical. But exposure to a chemical, in this case chemical X, causes so many mutations that all of a sudden 20 cells are going to revert instead of 2. So that tells you the chemical is most likely a mutagen. So in this case, this will be an indication that chemical is a mutagen. Now, this is the second part of the story. Probably it's suspected of being a mutagen. Now we can take the chemical and do other tests with it to see that if it indeed is a chemical, not just suspected of being a chemical, a mutagen. Okay, now that's one part of the story. The other part of the story would be that we are testing these with bacteria. Bacteria have no livers. The liver, what sometimes the liver does is it may take a chemical that is not a mutagen, so now let's say chemical Y, and it may convert it into a mutagen. So we test with the Ames test, chemical Y, then we plate our cells and we see how many colonies grow. Let's say 3 colonies grew. So these are 3 revertants. The control, we did it last time without the chemical, gave us 2 spontaneous revertants. So there's not a significant increase in number of revertants, which leads to believe that the chemical is not a mutagen. But what if the chemical may become a mutagen inside an animal that has a liver that will change the chemical into a mutagen? So what we do to test for that is we give the cells the chemical, and we also add liver enzymes. And now we test to see what happens in that instance. Not sure what. That's one of the liver enzymes. There's many. Actually, what they do, that's one of them. Yeah. What they do is they take actual liver. They take a little rat and they sacrifice the rat. They take the liver and they completely, actually, they put it into a blender. And that's what they put in there. That's how they get the liver enzymes. Yes, they do sacrifice rats. Yes, they do. Yeah, the liver has to come from somewhere. I'm sorry. Yeah, poor little rats. I know. Yes, that's the easiest way to do it. Just blend the liver. I mean, maybe nowadays you can actually buy already blended liver enzymes and just add it. But in my time, you actually had to sacrifice the rat and blend the liver. Yeah. So, yeah, that just, in the name of science, I guess. So with the liver enzymes, let's say now you get 20 colonies. Now we have a different story. Now you can say, oh, chemical Y may not be a mutant to bacteria, but it may be a mutant to an animal or a mammal. So to an animal with a liver that. Okay. So that's an added, something else that was added to the Ames test to carry it further to see whether non-mutagens to bacteria could become mutagens in a human. And there are many compounds that are changed by the liver into mutagens. So very, very good test, very easy, inexpensive. To date, there has never been a compound that the Ames test called negative that later was proven to be a mutagen. So it has an impeccable record. It has not ever given us a false negative. Yes, pretty much all of these is done first with the Ames test. So the Ames test is a screening test. If a chemical passes the Ames test, then it's safe because we have never yet found a false negative. It has never given us a false negative. So if the Ames test tells us it's negative, it's negative. If the Ames test tells us it's positive, then we could take it into further tests, and sometimes the positive may turn to be a negative, but that's okay. We can live with that. Or sometimes the positive turns out to be a true positive. So then we can take it into more expensive, more sophisticated tests with actual, animals. That may take years to see what happens to the animal over time, etc. So it's a very good screening test. That's what it is. And pretty much every single chemical that tests, we do it. It's tested by the Ames test first before it's taken on to more complex tests. So have an idea of how the Ames test works. So that if you're given the result of an Ames test, you can say, oh yeah, this is the interpretation of the result. Okay. So control plate 5 colonies, test plate 7 colonies, test plate with liver enzymes 15 colonies. Really 5 and 7 are pretty much the same number. 2 colonies more or less is not significant. Obviously 5 to 15, that's significant. That's 3 times more. So that's definitely significant. Okay, so now we get to the last part. This last part of the chapter is going to look into horizontal gene transfer. And there are 3 mechanisms of horizontal gene transfer: transformation, transduction, and conjugation. Your job is to keep them straight and do not confuse the T words: transcription, translation, transformation, transduction. So be aware of all those T words. Nice chart that summarizes all 3 mechanisms. And we'll start with the first one, which is probably the first one that was discovered, which is transformation. So transformation is going to transfer naked DNA, meaning DNA that has escaped a cell or it's without a cell, it's in the environment, and it's going to find its way inside another cell. Now, cells do not take up DNA. That's not something that they do unless exactly, like after a cell lysis, exactly. That's exactly what happened. After a cell dies and ruptures, now the DNA is out in the environment. And a cell normally will never take foreign DNA unless the cell is made competent, meaning that the cell may have weakened membranes, weakened cell wall, maybe pores in the cell wall, and the DNA manages to go in. So competent cells are able to take up DNA from the environment. We can attain competency. We can force a cell to become competent. That's very useful in the laboratory. However, in the natural world, transformation is extremely rare, very, very rare. This is not something cells do. They do not willingly take DNA. So the story behind transformation is actually quite fascinating, and it is fortuitous that it was discovered. Yes, competency means that the cells have a weakened cell wall, that the cell wall has become weak, it has been altered, and now it has pores large enough for a DNA molecule to go in. DNA molecules are huge, so it has to be a very damaged cell wall. will allow for DNA to go through. In the laboratory, we weakened cell walls, we make them weak by either shocking them with electricity or putting them in very high salt concentrations. That will make the cell competent. So those are two tricks that we do to make the cells competent. Turns out there's a bacteria that in nature sometimes makes itself competent, whether it's just because it's very sensitive to the environment and alterations in the environment weaken its cell wall. For whatever reason, Streptococcus is one bacteria that is known to be competent sometimes in the natural environment. Okay, so that's just one. Most bacteria will not do these, but turns out back in the 1940s, there was a microbiologist named Griffith who was commissioned by the British government. He was British, to create a vaccine against pneumonia, Streptococcus pneumonia. So he thought, cool, that's fairly easy. So they already had the bacteria that caused pneumonia. They already knew that one strain of the bacteria had capsules, and that was the pathogenic one. The strain of the bacteria that didn't have capsules was not pathogenic. He called the non-encapsulated bacteria ROF. The encapsulated one made a smooth colony, so he called it S. So he tested to make sure that indeed that was true. He put the S bacteria into a mouse. The mouse died. He gave the R bacteria into a mouse. The mouse did live. No effect. Okay. Then he took the S bacteria and he exposed it to heat and it killed it. So now it's dead. Gives it to the mouse lives. No problem. That's completely expected. Oops. Yeah. Okay. Then he did the next thing. This is the trick. He took the heated, killed S bacteria and mixed it with the live R bacteria. His thought is that's the vaccine right there. We're going to mix a dead bad one with a live good one, and now the immune system will learn to fight the bacteria. To his surprise, the mouse dies. And furthermore, when he goes into the mouse and does a blood culture to see what killed the mouse, the only thing he found was S bacteria. So now he had two possibilities. The dead bacteria came back to life, or the live bacteria was transformed into an S bacteria. The R transformed into an S. So keep in mind, he was working at a time when it had not been figured out conclusively that DNA was the genetic material. So he couldn't say, oh, the R took on the genetic material of the S and turned into an S. He couldn't say that. So what he said was that there must have been some transforming molecules from the S that were left in the environment, found their way into R and transformed R into S. So that's why he called it transformation, because he didn't know about DNA. It was still controversial whether the genetic material was DNA or if it was proteins. However, this experiment, once it was published, it piqued the curiosity of other scientists, and based on that, they began working and figuring out that it was DNA that was being the genetic material, not proteins. So that's transformation. First thing about transformation, extremely rare. It doesn't happen in the environment. It was just fortuitous that Griffith happened to be working with a bacteria, one of the few that we know of, that can naturally transform, that can naturally become competent and go through transformation. Okay. Nowadays, we do transformation as a tool in a laboratory to force bacteria to take on DNA. So it's become a really good technique. Oh, I have no idea why they call it competence. Yeah, instead of compromise. Yeah, you're right. Compromise would have been a better term. I have no idea why they called it competent. Yeah, I don't know why they chose that term. What was their thinking? Yeah, honestly, that's a good point, but I don't know why they did that. So In a streptococcus, the gene for streptomycin resistant, for example, happens to be acquired through competency. That we kind of figured that out. So one of the things that happens is that the DNA that goes in is a single-stranded DNA. So it goes into the cell, and DNA polymerase immediately takes it and makes the double strand, and then the gene inserts itself into the chromosomal DNA. So that's something about competency too, is that the gene that's acquired inserts itself into, it's not a plasmid. It becomes part of the chromosomal DNA. So at least in some instances, we know that's how some bacteria become either resistant to antibiotics or, in this case, pathogenic. Okay, so that's one, which again, very rare. This next form of horizontal gene transfer is not rare at all. It is very, very, very rare. The way this one works, this one is called transduction. And it requires a virus that can infect bacteria, which is called a bacteriophage, sometimes just phage or phage for short. Virus is what's going to transfer DNA from one bacteria to another. There are two forms of transduction, specialized and generalized. We're going to look at generalized. In chapter 13, we're going to look at specialized. So this is how it works. We have a bacteria, and this is a bacteriophage that is going to infect the bacteria. When viruses infect bacteria, they inject, just because he's standing next to, oh, I have no idea what you're talking about. Okay, so the DNA is injected into the cell, and now the DNA of the virus begins to transcribe and translate proteins that are going to destroy the DNA of the bacteria. So we have pieces of DNA of the bacteria that are being completely destroyed, and the virus will use those pieces, those nucleotides, to replicate its own DNA. So that's what it's supposed to happen. It's supposed to be using that DNA to replicate itself. What if, by accident, little pieces of bacterial DNA are not destroyed and are left behind? When the virus begins to replicate itself, it is going to encapsulate into the viral capsid the bacterial DNA. And now we have one little viral particle that, instead of carrying viral DNA, is carrying bacterial DNA. This whole cell ruptures. The other little viruses go off to infect other bacteria with viral DNA. One little virus, in some ways, yeah, like a mosquito, and one little virus will infect a new bacteria, but it's going to insert bacterial DNA instead of viral DNA. The bacterial DNA comes and joins with the chromosomal DNA of the new bacteria, and now that new bacteria has been transduced. It has acquired new DNA. This happens all the time. This is incredibly common, almost scary how common it is. Most of the organisms that make toxins, they have acquired these toxins through transduction. Through these phages that are transferring these genes for the toxins between one cell and another, more so than any other mechanism. So, very, very common way of acquiring new genes. All right, so that's basically explaining the same thing. And the last one is conjugation. Conjugation is, again, it's common. This one requires a plasmid. And we mentioned plasmids in chapter 3, and it talked about plasmids being little pieces of circular DNA that bacteria have. And the thing about plasmids is that they're incredibly versatile. A plasmid can join with other plasmids. Okay. A plasmid can go into the DNA and be part of the DNA, or it can pull out and be back to being individual plasmids. There are plasmids that carry resistance to antibiotics. the plasmids we're going to talk about are called fertility plasmids, which allow the bacteria to create a conjugation pilus with which they gain access to other bacteria and they transfer pieces of DNA from one to the other. So essentially what happens is one bacteria has an F plasmid. The F plasmid allows the bacteria to make a conjugation pilus and connect with an F minus bacteria. And that's something that to note is that F positive bacteria only connect with F minus bacteria. Once that happens, the plasmid, a single-stranded piece of the plasmid begins to be transferred to the new cell. The recipient cell creates the second strand, so now we have again double-stranded DNA. Both cells end up with double-stranded copies of the F plasmid, and now both cells are F plus. So the F minus was transformed into an F plus. Okay, so that's good. Good question. Do the bacteria have to be the same genus and species? No, they don't. And that is the amazing thing, is that you can have two bacteria from completely different genus and species, and conjugation can happen between them. So you can see how genes from very different bacteria can be transferred. Right. That is the amazing thing. Correct. Yeah. Now, this is the other deal. Now, you would say, okay, so who cares that they do these? I mean, all they're doing is they're transferring the F plasmid. And now this bacteria can make a little F conjugation pilus and connect to other bacteria. I mean, that's nothing to us. If that's what they want to do, that's not harming us, unless two things that could happen. But now we're bothered by these. One of the things that could happen is that the F plasmid could go into the chromosome of the DNA of the bacteria. We call that an HFR. So when the plasmid goes into the bacteria, now the bacteria is not an F plus cell anymore. We call the cell an HFR, just to say, okay, now the plasmid is into the chromosome of the bacteria. Then the F plasmid create or codes for the pilus. It goes into the other bacteria, connects with the other bacteria, and the origin of replication of the plasmid will be right here. It begins to put out a piece of the DNA. But because the transfer began at the middle of the plasmid, it will continue on and it will begin to transfer part of the chromosomal DNA. So we have part plasmid, part chromosomal DNA that is being transferred. These connections do not last very long, so the cell will cut off the connection, or the connection will cut off before more DNA, the entire genome is transferred. That's never going to happen, just a little piece is transferred. But now the new cell has part of the plasmid, and more importantly, part of the chromosomal DNA of the donor cell, which means these genes could be genes for toxins, genes for capsules. And now the new bacteria that perhaps did not have capsules or could not make the toxin have now acquired those genes. Okay. It could be genes for resistance to antibiotics, for example. So now we have an instance in which we care. So that's how conjugation can harm us, can create bacteria that are more pathogenic. That the transfer does not hurt either one of the cells. So it's either going to acquire genes that are not good or bad, or may acquire genes that makes the cell more competitive, more pathogenic. Okay. So no, the cell is not hurt. So that's essentially how conjugation works. And this last one just talks about vancomycin resistance in Staph aureus arose from conjugation between Staph aureus and Enterococcus. And basically the problem here was that vancomycin, turns out, is the antibiotic that they give to animals to make them grow bigger, quicker. And so Enterococcus, which is a normal flora of the colon, was exposed to low doses of vancomycin for a long time. And now it became, some of the resistant strains began to proliferate. And some of the resistant strains of Enterococcus faecalis was transferred by conjugation to Staph aureus. So now we have vancomycin-resistant Staph aureus, which is really bad because vancomycin was one of the few antibiotics that can still, in most instances, is supposed to destroy or kill Staph aureus. And now we have vancomycin-resistant Staph aureus. So that resistance came about through conjugation with Enterococcus faecalis. So what you need to know from these is keep all three events, all three mechanisms separate. Know which one requires a bacteriophage, which one requires a plasmid, which one can transfer chromosomal DNA, which one, I guess, transfer just pieces of DNA. Pretty much all of them just transfer pieces of DNA, actually. Okay. So keep a mystery as to which one is which and be able to answer questions about the mechanism. All right. Don't know why transduction is called transduction. Not sure what transduction. I think transduction means to change what? Like a transducer. A transduction. What's a transducer? A transducer is like a what is it? Somebody knows? Like in senses. Yeah, transduction changes. It's an interpretation of something. When we transduce a message from one thing into another thing. Maybe that's why they call it transduction. So the transduction of a message is kind of the changing of a message from one place to another. And maybe that's why they picked the word transduction. It was changing the message or transferring the message from one cell to another, the message being the DNA. Yeah, maybe that's where the name comes in. Yeah. Okay. Any questions? All right. So we should be ready for the next test. The next test one is a next week. Yeah. Okay. All right, guys. So we'll meet again. Yes, I'm gonna do that right now. I'm gonna put in lab 8 right now. Lab 8 goes with all this material, and lab 9 a little bit too. I can't remember now. Yes, lab 8 will help on this lecture test because lab 8 is what is it? Is a DNA. What is lab 8? What is it called? It is biology. Is DNA structure and technology. So you need to know about DNA structure. So it's definitely going to help you with that. DNA biology and technology. Actually, 9 is the one that has. What does it have? It has one of these mechanisms. I can't remember. It has transformation, I think it is, or transduction. I can't remember which one it has. Let me take a look it up. Yeah, 9 has one of these mechanisms. Okay. All right, guys, I'll go ahead and add those labs so that they'll be available. And we're wrapping up labs very quickly here. The 7, 8, 9, 10, and that's basically it. So we have 4 more labs to go. All right, have a good night, and we'll see you Monday for lab.